But now to measles cases climbing once again in parts of the world. And now researchers here in Atlanta say they may be getting closer to a pill that could actually treat infections caused by measles and similar viruses. Let's talk more about it now with our next guest, Dr. Richard Plemper from Georgia State University, one of those researchers. Dr. Plemper, it's great to have you on the show. Thank you for your time, sir.
Thank you very much for having me.
Okay, so we're talking about this new potential oral drug. It's GHP 88310. How does it work against these viruses?
So measles is a virus that has an RNA genome, and that is a specific polymerase that the virus needs to replicate this genome, so to amplify itself. And this drug targets the polymerase complex, which is very specific to the virus or to this family of viruses, and blocks its activity. So that means the virus cannot replicate its genome any longer, and so therefore cannot produce any progeny particles. So it interrupts the transmission, the viral replication stage at the genome amplification level.
In addition to the measles, what other viruses could this potentially treat?
Yeah, measles belongs to a subfamily of viruses that includes some of the major human and also animal pathogens. One major group of this in this family are human parainfluenza viruses that, in particular in children, cause, for example, croup syndrome, but can be potentially life-threatening with viral pneumonia in immunocompromised and in particular in transplant patients. And so that was actually our original target group, patient group for this drug, were high-risk transplant patients infected with the parainfluenza virus. And what we then discovered once we identified this drug class and started to characterize it was that its activity was actually broader, so it doesn't only block the parainfluenza viruses, but it also blocks measles virus and related animal viruses that are very similar to measles, like, for example, canine distemper virus that causes distemper in dogs and is in particular very, very dangerous for the dog owners in puppies if they are not vaccinated.
You know, as for the measles, we're seeing some of these outbreaks in the U.S. right now. Reams, correct me if I'm wrong, I believe we're up to like 5 cases here in Georgia that have been confirmed so far this year. I mean, what would these outbreaks look like today if this drug were readily available right now?
Yeah. So at the moment, the heat of the outbreak in the last few months was more in Utah, and you may remember later last year we had a hotspot in South Carolina, and it all started in earlier 25 with the outbreaks in West Texas. But it's really not only limited to the United States. We have at the moment more than 17,000 measles cases recorded in the last few months in Mexico, and also very large outbreaks in 25, 26 in Canada. So it's really not regionally contained. It's a broader North America measles epidemic that we are looking at. And so traditionally, how we have controlled measles is through ring vaccination. Mm-hmm. If you have an index case, then you vaccinate as fast as you can all potential social contacts. So this is, for example, in daycare centers or schools, you identify the contacts of your infected child. And by doing so, you try to regionally contain the virus and ultimately silence the outbreak. Now, if vaccination is declined, so if ring vaccination is simply not applicable because of hesitance of caregivers, then we run into a situation as we experience right now that an outbreak is not contained but becomes larger and spreads over large geographical areas. And what we think is this a drug like what we develop can augment vaccination and actually step in if ring vaccination is declined. One should treat contacts, and this can be as a pre-exposure prophylaxis or even as a post-exposure prophylaxis. So a child is already infected, but has not yet developed any clinical signs of measles, like fever and then the rash and overall severe disease. So one can treat pre or post-exposure prophylactic, or even, as we have demonstrated in animal models, also therapeutically. And by doing so, pre and post-exposure prophylaxis completely silences the virus, meaning those patients, again, it's based on animal model data so far, but those contacts do not develop any disease. and therapeutic treatments, so after the onset of clinical signs, if started still reasonably early, greatly improves outcome. So
Wow.
disease shorter, and also for further spread. So is there epidemiological potential benefit? The animals also stop shedding the virus sooner when treated, and that means if a patient does not shed the virus, then of course it also cannot transmit. So again, that contributes to silencing outbreaks.
Dr. Plemper, you're doing a great job explaining everything. You'll have to forgive me, but the extent of my biology study is 10th grade of high school. So we might need to dissect this a little more. I'm trying to understand, and when it comes to measles specifically, this isn't to replace the vaccine, right? But this can step in and help with treatment for folks who get it.
Right. So this is definitely not to replace the vaccine, but what we have seen is that if the overall goal is really global measles eradication, and I feel this should be the goal because we believe it's actually feasible, then can be reached this with vaccination alone. And as it looks at the moment, in theoretically, it's possible, but right now the numbers do not look so encouraging. And so therefore we think, how can we actually augment vaccination towards this ultimate goal of global eradication? And that is, we find, is where a therapeutic and orally available, so a therapeutic that can be taken by mouth, comes in, like the one we are developing, so like GHB 88310, that again can contribute together with vaccination to silence an outbreak early. And again, if transmission chains are interrupted, as we've seen in the pandemic, that is what needs to be achieved to stop a virus in its track.
Dr. Plemper, what's the next step for you guys? I was reading through the study this morning on science.org. I saw a lot of it was tested in lab rodents. What does this mean
Yes.
for potential human clinical trials and hopefully one day getting before the FDA?
Yes. So at the moment, this drug has been tested as you said, in animal species, in rodents, but also in non-rodents. So we tested safety in ferrets and also in dogs, and our efficacy model is also a ferret model. And so essentially, it has completed all of the experimental phase testing. And the next stage, as you said, to achieve approval, FDA approval to start a clinical trial, is what we call formal preclinical development. That's essentially very standardized testing of safety of the drug to determine at what concentration can one safely go first into humans in a clinical trial. And so that is the next stage, formal preclinical development. And if it passes all the required thresholds, the milestones, then clinical testing would start first with a safety trial, and then with a trial that tests effectiveness.
And any idea on the timeline for that?
That's always the question. So the fastest we've ever seen a drug advance from the state where we are currently, that was at the onset of the COVID-19 pandemic. The drug that became molnupiravir was about at the stage when the pandemic started, where we are now, and so we contributed quite a bit to the early work of molnupiravir development with our lab. And that timeline was about unprecedentedly fast and give or take 2 years. And now measles, one cannot, although the clinical need is major. I mean, measles is not a benign disease. And we have many case fatalities in Mexico and several in the United States. So one needs to take it very serious. But clearly, it can still not be compared to the onset of the SARS-CoV-2 pandemic. So realistically, we are talking several years.
All right. That's Dr. Richard Plemper from Georgia State University. Great work by you and your fellow researchers there. Incredible to see this stuff being done in our own backyard. I mean, they're calling this the most promising inhibitor of this virus family they've seen in years of research. So kudos to you and your team. Thank you for your time, sir.
Thank you very much.